and she brought this wind this morning. She brought the wind. So anyway, Professor Dorothy Hanck is a professor in the Department of Medicine, University of Chicago, and in particular in the Division of Cardiology. And Dottie, as she's often called, graduate, got a Ph.D. from Temple University in Philadelphia, and then had postdoctoral fellowship in Leeds, in the UK, from which he went to University of Chicago and started another postdoctoral fellowship with Harry Foser, and that was the end. She stayed there until now. She's not living. Harry's now retired, and I think he spends most of his time in North Carolina, where he tells me he has a beautiful home with a beautiful orchard. So that's I've never been. Dottie, we're happy to have you with us.
Well, thank you very much for the invitation. I'm going to try to turn this on. Okay. All right. I'm actually very delighted to be here today. I want to tell you about something I've been working on for a really, really long time. And sometimes that's kind of embarrassing. Students often ask me, don't you have it figured out yet? And I'm really happy to report that I think we almost have it figured out. This class of drugs has been around for almost 70 years. The prototypical drug lidocaine is still used in the hospital setting. And this class of drugs has a blocking mechanism that I would say is the template against which all drug interactions are first tested. And so my excuse is, if we can really understand this class, then we're a lot closer to understanding a lot of other drugs that do the same thing, but with just a little bit of variation on a theme. So today I want to focus on something that I hope this group is very sympathetic to, and that is the idea that local anesthetics are a nice, large class of drugs, but what is it that makes a local anesthetic grow up to be an antiarrhythmic drug? And the work that I'm going to tell you goes back a long way. John Mikulski, who has been at the University of Wisconsin now for a long time, was involved in some of the very early experiments. Mike Sheets is involved in virtually everything I'm going to tell you about today. He was originally at Chicago. He and I were postdocs together. We continued to work together when he went to Northwestern, and we've continued to work since he moved to Utah. And hopefully, if the NIH is kind to us, we're going to keep working together for the foreseeable future. And this array of people, including Harry, Gregory Lipkin, who does all the molecular modeling for our group, and some graduate students, some technical people, and a postdoc, also contributed to various parts of the work. So the class of molecules that I'm going to talk about, of course, lidocaine is the prototype, but today I'm going to tell you both about lidocaine and this drug that's used as a local anesthetic, but is a really rotten antiarrhythmic drug, benzocaine. I'm going to try to convince you what the difference is between these two drugs. And then I'm not going to talk about the QX drugs today, but what I'd like you to notice is that sodium is really small. It travels around with a little shell of water around it, and this is size-wise not too terribly different from the ring structure of benzocaine and lidocaine. There are a whole bunch of drugs in the same cane family, many of which are used as local anesthetics, and a subset of which actually also work as antiarrhythmic drugs. And of course, cocaine itself is in this group as well, although it is neither a local anesthetic nor an antiarrhythmic drug. So anyone, probably I don't need to show this to this audience, but like many speakers, I'm going to talk about it anyway. We've known since the very nice work out of the Ketterall lab in 1994 that there are amino acids on the domain 4S6 of the voltage-gated sodium channel, which when mutated decrease the affinity of lidocaine-like family drugs. The most important of which, and why I say the most important, is the largest change in affinity was seen when this phenylalanine here, just below the selectivity filter, which would be right about here, was substituted with an alanine, and this tyrosine. And then later on, some data showing that there was a leucine over in domain 3, and there have been a couple of other amino acids in random places, but the most important of which, and by that I mean the largest changes in affinity, have been noted for this group of amino acids. In 2005, Gregory Lipkin and Harry put together a model based on the open and activated structure of the MTHK potassium channel, and then it's a homology model, so they took the domain 4S6 and domain 3S6, substituted the amino acids from MTHK with those of the sodium channel, minimized them so that things didn't bump into each other, and docked lidocaine and lidocaine-like drugs in this binding site, utilizing all of the mutagenesis data that was available before 2005. And this included people who did alanine substitutions in all of the S6 domains of the S6 segments of all 4 domains, as well as a variety of people who substituted with other amino acids like tryptophan or lysine or a number of other things. And at that time, really none of our own work that had come from a variety of labs, a variety of isoforms, and Gregory docked the molecule and said all of the data that are available are consistent with the idea that the amino end of lidocaine makes pi electron interactions with this phenylalanine 1759, that there's an aromatic interaction that happens between the ring of lidocaine and tyrosine, and the whole thing sits wedged between domain 4S6 and domain 3S6 with this leucine, the one that had been shown to be playing an important kind of ledge role in making the binding site. So our idea now was to try to reconcile this idea with the actual electrophysiological data that were available. And this figure is taken from our paper last year in J. Physiol, in which we looked at some electrostatic interactions. But for purposes of today, I want to introduce the nomenclature. I want to impress you with the fact that one of the problems we have with electrophysiological data is that we've come up with a whole vocabulary for talking about how drugs interact with channels based on electrophysiological data and various protocols, and we in part can reconcile that with simple Markovian models of how the channel might work, and to a lesser extent with structural homology model data that we have of the channels. And our goal, as I've learned today in Jorm's lab, your goal, some of you, is to try to reconcile, like we are trying to do, the more structural data that we have with some of these ideas that we have developed over the years. So some of the nomenclature that's out there that I'm sure you recognize is the fact that if you take a cell under voltage clamp and you expose it to drug while holding the channel at a very negative potential And then the very, very first time you depolarize it, if you have a drug like lidocaine on board, some amount of the current is gone the very, very first time you depolarize. This is usually called first pulse block. I like to think of it like a first kiss. During the time that you're allowing things to equilibrate, channels are living mostly down here in these deep closed states, and only very, very rarely, if at all, do they experience any of these states over here. And so this is the time when you get the best idea of what a closed state interaction, especially if you have an extremely negative holding potential. So channels live mostly down here in these deep states. Anything you do after that, no matter how long you wait, is going to now involve some number of channels that have visited these different states in some kind of equilibration with as being drug bound. And certainly if we pulse the channel quite rapidly, in this case a very common mechanism is to use something like a 10 hertz train. After a 10 hertz train, virtually all the current is gone. And the amount of current that we would then measure in the presence of some amount of drug, we would call tonic block. That's the kind of block you get that represents the steady state amount of block at whatever the resting potential that you have, but the idea is that this amount of block is always greater than this first pulse block because you can only be kissed once. We can do dose response curves then for all of these different kinds of block. First pulse block gives us the lowest affinity interaction. And here for lidocaine at room temperature under these particular experimental conditions, the KD is about 1 millimolar. Use-dependent block under most conditions, this open and activated state block is generally reported to be in the range of 10 to 20 micromolar under a variety of different conditions. And tonic block is going to be someplace in between here, depending upon how you actually do the experiment. And I'm going to show you some data later on to convince you of that further. Now, what we really want to know is what is it that's different? Is there anything different about the kind of block that you get in tonic block from the kind of block you get when you pulse rapidly or you depolarize for long periods of time? And to answer that question, we turn to the idea that voltage-dependent block, by definition, seems to have to involve voltage sensors, since either that or a drug has to actually bind in the field. And there really have been no data to support the idea that molecules like lidocaine, for example, really have a strong voltage dependence by just moving through the field. This is separate maybe from the QX molecules, for which you can show that there is some voltage-dependent flicker block, but the affinity of that block is in the multi-millimolar range and unlikely to be responsible for the voltage-dependent block with an affinity that's down in 100-fold lower, or at about 10 micromolar. So many years ago, Mike Sheets developed this wonderful mechanism, which we first used in cardiac Purkinje cells and now have adapted for use with HEK293 cells or heterologous expression in other cells, by which instead of measuring flux through channels by measuring ionic current, instead we measure the movement of the voltage sensors directly by replacing all of the permeant ions and measuring only the charge movement associated with them. These are some typical recordings under the conditions we typically work these days. We use this very, very large pipette. The aperture in this pipette is about 30 microns across. It's huge. And in fact, we use the same pipette over and over and over again. Mike and I, in the 23 years we have worked together, have, I believe, only used 3 pipettes. And one of those only lived for a very short duration of time. And so most of the data that I show you from, that Mike I've generated, comes from one of 2 pipettes.
What are their names?
So here, in order to take advantage of an aperture that's 30 microns across, obviously some itty-bitty HEK293 cell is not up to the job. So we fuse the cells together by using polyethylene glycol. We just kind of mush them, expose them to it, mush them together, and over a period of a couple of days, they remodel their membrane. That is, they get rid of all the extra membrane, and they make these big round cells. And you can see here, if you aren't too far in the back, that there's 2 regular cells kind of stuck onto this big cell, but they're not electrically coupled to it, I don't believe. We picked this one so you could see the difference between the size of the fused cell and the size of the individual cells. In this particular case, these cells were 400 picofarads as opposed to, say, 10 to 20 picofarads that a typical HEK293 cell is. But we study cells as big as we need to in order to be able to get enough channels so that we can generate gating currents that are multiple nanoamps in height, maybe as tall as 5 or 10 nanoamps in height. And the reason we need them to be so big is that we have a very wide bandwidth. We record with a bandwidth greater than 100 kilohertz. So the signal to noise, signal has to be pretty big to be able to record the signals in the presence of the kind of noise you get in that thing. So you do this experiment, and what you find is that unlike control gating currents, in the presence of a large concentration of drug, which you choose in order to be sure that absolutely every ion channel has a drug associated with it, that the gating currents are much faster and shorter than they were in control. And you can see here that the decay is very much less voltage dependent than in control, and the amplitude is shorter. As well, and probably more importantly, the amount of charge that you see, and these are actually lidocaine data, that there's much less charge passed at positive potentials. The overall slope factor is less, and charge appears at negative potentials that wasn't there before. So there's some very important gating changes that lidocaine introduces, and lidocaine-like drugs introduce into the gating of sodium channels. In point of fact, as you change the concentration of drug and modify the fraction of blocked or bound channels to unbound channels, what you find is that these changes are dose dependent. So here, for example, with a lower concentration of lidocaine, you get some amount of charge that's gone in the first pulse, and then as you develop a train of depolarizations, charge further goes down. This steady amount of charge reduction is shown here with a KD of about 20 micromolar for total charge reduction, and in this case, the tonic block is at around 200 micromolar. And this compares very well with the KDs or ED50s that you measure when you do the same experiment using ionic current. Here, for example, the tonic block is shown here, and the total block again is about 20 micromolar, and the tonic is at about 150 micromolar. So it looks like a duck, it walks like a duck, it looks like the voltage, there's some tonic amount of charge reduction, and so it says that the voltage sensors are involved. And in fact, after that, we went and did a series of experiments in which we tried to figure out which parts of the channel were attacked by the drug. How was drug binding in the pore of the channel? Because clearly we, I think, all agree that these drugs are affecting the channel by binding in the pore. What voltage sensors were affected? And I don't have time to tell you about all these data that have now been in the literature for about 5 years, but we did a bunch of mutagenesis experiments in order to be able to identify where the charge came from, and those experiments showed that about 40% of the charge, the overall control charge of the channel, comes from gating of domain 1 and domain 2. We were not able to distinguish these two from each other. About 30% of the overall gating charge comes from domain 3, and about 30% from domain 4. In the presence of lidocaine, domain 1 and domain 2 gate normally. They seem unaffected. Domain 3, on the other hand, is missing entirely. Domain 3 refuses to move any longer when drug is bound. And the domain 4 charge moves, but it moves not as much as it used to, and it's actually the charge that moves down here. It's domain 4 charge that gets augmented in the presence of drug. In this set of experiments, we also determined that the domain 3 voltage sensor is actually stabilized in the outward position, as if it's being held, that it's being held up artificially by lidocaine.
So the essence of the experiment is that domain 3 mutations don't affect charge movement in the presence of lidocaine.
Right. Mm-hmm. That's right. You have less overall charge, but then it's not affected by lidocaine. So that brought us to the next point, which is trying to figure out how do ionic current and gating data actually arise from these drug channel interactions that have been proposed based on all of the mutagenesis data. So now I have to, that's where I'm going to take you. And so here are this set of mutagenesis. We focused on phenylalanine 1759 because it produces the largest change in drug affinity of any of the mutations, and because in NAV 1.5, that is the cardiac version of the sodium channel, that when you mutate that phenylalanine to a whole variety of different amino acids, including alanine, cysteine, you can make it negative with aspartate or glutamate. You can put a positive charge by putting arginine or lysine there. You get a common phenotype. The common phenotype is that in the presence of 1 millimolar lidocaine, the affinity that you get of the channel is not different based on what you put there. And moreover, when you pulse at 10 hertz, you get no additional block. So use-dependent block, the hallmark of making an antiarrhythmic drug, is abolished in every single mutation. Now there are some, where at this time there were already data in the literature in another isoform of the channel that said had we put another ring structured amino acid at this position, we would have retained some use dependence, but for purposes of this particular study, we did not do that. But we were very intrigued by the fact that this first pulse block did not shift down to 20 micromolar, and in fact, by these assays, it would appear that we eliminated voltage-dependent or certainly use-dependent block. So we chose two of these to work with, F1759K, which puts a positive charge at this position and would be expected to very nicely antagonize the positive charge on the amine of lidocaine, and 1759A, a neutral substitution, which also in this assay does not produce use-dependent block, and is, pretty small. And then we wanted to do some gating current experiments, which we typically do at a very cold temperature. So we cool the preparation and then ask the question, well, how do these mutant channels behave in this very cool temperature that we typically do our gating current measurements at? And first, this is F1759K, and what I'd like you to notice is that one of these is 1 hertz, and the other one is 10 hertz. The channel inactivates a little bit in a train at 10 hertz. You can see that both of these channels do that, as does a wild-type channel. And in the presence of, in this case, a millimole of lidocaine, here's your first pulse block, and then the 1 hertz train, and your 10 hertz train. So in the cold, even wild-type channels show a little more use dependence in the cold at lower frequencies than they do at room temperature. But F1759K is totally not use dependent, but its rested state, to try to use the terminology in the literature, or closed state, or tonic block, is unchanged and has a KD or an ED50 that's only a little bit higher than that of the wild-type channel. F1759 has a whiff of use dependence, very, very small. The ED50 and 10 hertz trains is improved only by less than a factor of 2. Remember that lidocaine itself in a 10 hertz train will improve the ED50 50 to 100-fold. So the interaction, supposedly now, these reagents would allow us to look at whether we can separate out the voltage-dependent interaction of lidocaine from the voltage-independent action. And that's exactly what happens. In the 1759 channel, you put on 10 millimolar lidocaine and absolutely nothing happens. Remember that in the control channel, you induce this extremely dramatic change, which is fully reversible upon washout of the drug. 1759, in keeping with the fact that we saw this very, very hint of use dependence, produces maybe some alteration if you're, really want to believe that there's voltage dependence here. You could say that there was some effect on the voltage sensors with this this mutation. So, so this then, just go back here. This then confirms, or if you will, separates out, or potentially is a tool to tell us that there are two completely different kinds of effects that the lidocaine and lidocaine, maybe like drugs, are having with the channel. One is a simple voltage-independent plug, because these channels also get blocked by lidocaine, but the KD ED50 is fairly high. And then there is some voltage-dependent interaction which is orchestrated by this lysine. And so now we wanted to go back to ionic current data and say, okay, now if we understand that there's really just two things that happen, just two, can we see this in the ionic current data? And the answer is, of course, yes, or I wouldn't be here. And so the protocol that is most telling of this interaction is a steady state availability protocol, whereby you change the holding potential for some duration and then test to some potential that opens as many channels as are going to open. And under control conditions, you get a steady state availability curve. And then you choose some holding potential that's so negative that you're well away from where this voltage dependence reduction occurs, and you expose to drug. And when you do that, you find that there's some steady level, which is equivalent to tonic block, that you get, and then you get some range of potentials over which the current increases, and if you scale this up, it appears that the steady state availability relationship has shifted to the left. And this shift is taken to represent the fact that you have some voltage-dependent dissociation of the drug with the channel, and as you get more negative, the voltage-dependent block is reduced until finally you get to this tonic block state. Now, that's a very standard sort of idea of these kinds of data in the literature. Of course, the real problem is that if you hold the cell at a different potential, one that's even more negative, what you find is that the availability relationship still has the same general form, but now it doesn't shift as far to the left, and the actual estimate of tonic block is not as great as it was when you had this potential holding it. In the control case, there was no reason not to choose this potential. It was well negative to the range in which channels inactivated. It was only after you applied drug that this became an at-risk potential. So in the limit, if we were to hyperpolarize farther and farther and farther negative, one would imagine that eventually one would come to some final resting value of tonic block that minimized interactions that were producing this reduction. And I want to argue to you that this reduction in tonic block, or the kind of tonic block numbers that we have been accustomed to looking at in the literature all of these years, are a big mixture of the voltage-independent block that lidocaine and lidocaine-like drugs have, and the voltage-dependent block. In other words, after in these experiments, there are always channels trapped, if you will, or bound to various other states besides the deepest closed state. And it isn't just rested state block. And that really comes home when you look at the mutational data for F1759K and F1759A. Here is the wild type channel at now the very cold temperature, where the voltage dependent ED50, we held at a really negative potential for these experiments down here to try to get, and what you see is this little shelf appearing as the time constants of unblock of the various states now compete with the time course of our conditioning steps. And this is absolutely missing in the 1759K mutation, where the shift in voltage-dependent availability is non-existent, yet you still get this very nice reduction, or what we would have typically called tonic block, but which now I would like to call voltage-independent block, or as I'm going to try to convince you from some modeling, lipophilic block. And this ED50 is in the same range as the first pulse block that we would see to the deepest closed states. F1759A, on the other hand, brings up a very important point that I want to transition with you to another drug, benzocaine. This drug was barely use-dependent at all at cold temperatures, and at room temperatures, it had no use dependence whatsoever. Yet in this availability assay, what I'd like you to notice is that it does a really fairly good job at shifting steady state availability. Of course, recognize that these concentrations of lidocaine are really pretty high, 5 and 20 millimolar. But it's doing a fairly good job. Obviously, there are some conditions under which voltage dependence is evident, although the time course must not be such that they reveal themselves in use-dependent block. And that then caused us to consider the case of benzocaine. Benzocaine is well known not to be a voltage-dependent blocker. And in fact, there are some rather confusing data in the literature because in this availability assay, even though the channel is not use-dependent, it has a very marked propensity towards shifting voltage-dependent availability in the negative direction, suggesting that benzocaine itself can produce voltage-dependent block. Well, uncharged benzocaine is very difficult to get into solution. In fact, you can't get more than about 2 millimolar of it to actually dissolve in water. And it's one of those uncharged molecules that if you try to trick it by putting it in DMSO and shaking it up and then putting it in your solution and praying that it doesn't come out, that it does. It comes out really quite quickly. And so anytime you read a paper that says they studied benzocaine at higher than about 2 millimolar, don't believe it. They just didn't. So you put 2 millimolar benzocaine on, and that's as much as you can get. You can block quite a lot of the current. And you get a partial, uncharged benzocaine now, not the charged lidocaine, but the uncharged benzocaine produces a gating charge change very reminiscent of lidocaine, of a partially blocked lidocaine state. You just can't block all the channels by benzocaine to get a full charge. Interestingly, benzocaine goes on and comes off the channel so fast that you actually can watch block develop during a step depolarization. In this case, the red line is the depolarization in the presence of benzocaine, and the black is in the absence. And this little bar graph shows you the abbreviation in time to peak, that is, block of benzocaine occurring during the depolarization that then fully comes off in between depolarizations, and which is why it is not use dependent. On the other hand, you can see some effect of this, that it actually is voltage dependent in the 1759K channel. Like lidocaine, it no longer produces a shift in the availability relationship, even though it continues to be a very good tonic or lipophilic or voltage independent blocker. And in the gating charge assay, I will grant you that you probably do not find it surprising at all that 2 millimolar benzocaine does absolutely nothing that you can take home to the bank to the charge voltage relationship. Now, 1759A, on the other hand, does produce some shift in voltage-dependent availability, although much less than, say, lidocaine does. And it, again, continues to be voltage-dependently blocking the channel with an affinity predicted to be about 3 millimolar. So now, because benzocaine is so fast, and it's fast under all of the conditions that we've studied it, that means it isn't use-dependent under any circumstances, we can make a kind of assumption. We can say that it's reached some quasi-steady state, and we can therefore utilize the information about the shift in availability as being indicative of voltage-dependent block. We can't do this for lidocaine because lidocaine is use-dependent, which means by definition we are not at steady state. But for benzocaine, I think we can get away with this, and so we can say that here we have now this rested state or voltage-independent block by lidocaine, which is in the same order of magnitude for all of the channels. That for benzocaine, it's actually a better lipophilic blocker, and it only changes by a factor of about 2 in F1759K, and it goes up to 3 in F1759A. That we know that the voltage sensor block is about 20 micromolar. We've known that for years and years and years. But now we can make an estimate of what the voltage sensor block affinity for benzocaine is, utilizing this kind of analysis, where the data fall very nicely on a single site dose response curve. I'll grant you that it's not a strong argument in favor of the analysis technique, but it's encouraging, and it says that the affinity of benzocaine now for the voltage sensor is about 0.5 millimolar, as opposed to lidocaine, which has an affinity for the voltage sensor open and activated conformation of about 20 micromolar. So it's this difference in affinity, that makes benzocaine a lousy antiarrhythmic drug, when in fact it can interact with voltage sensors. And it accounts for the fact that this affinity and this affinity are so similar that in ionic current experiments alone, we can get confused about what's going on. Notice that the F1759A channel is predicted based on its shift to decrease affinity only a little bit less than an order of magnitude, where 1759K doesn't seem to have any ability to engage the voltage sensors. So how do we now put this together with what we think about, that we know about the binding site in the channel? This is the confirmation that Gregory and Harry worked with in 2005, and it's based on all of the mutagenesis data that was available up until that point. At that time, they didn't spend any time thinking about the closed channel because none of the mutagenesis data available really said that there was anything that you could mutate anywhere on any of the S6s that affected tonic block, except in a few cases where there might have been a change of about 2 or maybe at most 3, which now we would interpret to mean that they probably did the experiment under conditions where tonic block included some amount of voltage sensor block in it as well. And so they were a little misled in thinking that they had produced a change in rested state. So in this configuration, we start with lidocaine. This is the 2005 model. Lidocaine has its amine, making pi electron interactions with F1759, aromatic interactions with 1766, and it kind of sits on this leucine shoulder in domain 3. Now, the closed channel, so the open MTHK channel kind of looks like this. The closed channel of the KCSA channel has this bundle crossing at the bottom. If you look at that closed channel structure, inside are predicted to be only about 50 molecules of water. It's virtually impossible to, you cannot dock lidocaine in that conformation very effectively. And lidocaine and benzocaine displace about half of the water. So the inside of a closed voltage-dependent sodium channel is actually a very structured water condition. It's most unlikely that the dielectric constant of that body of water is 80. It's probably much lower than that. And it probably biases lidocaine to exist in its uncharged form. Because and most likely also explains why charged local anesthetics and antiarrhythmic drugs do not in general bind well to closed channels. QX molecules do not bind to closed channels. Flecainide with a pKa of 9.3 also does not bind very well in closed channels. Probably not because they don't fit very well, but because their propensity or greater propensity to be charged, in the QX case to be permanently charged, and in the flecainide case really, really, really wanting to be charged, makes it a much less hospitable condition for the drug. On the other hand though, you can orient lidocaine, instead of orienting it vertically as it sits in the open and activated conformation, you can tip it on its side, turn it around so that now its amine end, which is now neutral, faces away from 1759, and the aromatic ring faces towards the phenylalanine, making an aromatic interaction. Actually, then you can fit lidocaine into the channel along with about 22 molecules of water. And you can also then dock benzocaine in exactly the same configuration, so that in the closed state, we don't have as high an energy interaction because of this aromatic interaction. And also, if you take it away, because it's a generally more hydrophobic environment, there are lots of opportunities from all of the amino acids on the walls of the channel to contribute a little bit to the binding energy. And it's probably the reason why we've never found any closed state, important amino acids for determining closed state interactions. Now, interestingly, it would appear that whereas lidocaine will reorient itself so that its charged amine is up towards the phenylalanine, making pi electron interactions in the open and activated state, benzocaine probably sits in the same orientation with its ring structure up. We don't really normally think about benzocaine this way, but this end of benzocaine, which has a nitrogen right here, is actually an aniline structure. And as an aniline structure, and the fact that there's this electron withdrawing ring here, does make a very partial positive charge expected to be at this end of the molecule that has a nitrogen on it. that is not as strong a positive charge, but still a positive charge, like the positive charge that comes onto lidocaine. And so this can orient itself with the phenylalanine of 1759 in a similar sort of interaction that lidocaine makes, and therefore have benzocaine sit in, the open and activated state in the same general orientation as lidocaine does. Of course, we don't actually have any specific experimental data that say it's absolutely like this, but the data all fit this kind of idea, and they fit this kind of idea better than a whole host of other orientations actually fit. So, in summary, what I've tried to tell you about today is that local anesthetics and antiarrhythmic drugs actually bind the voltage-gated sodium channel in pretty much the same way. The difference is they interact with similar amino acids in the pore of the channel, and the only thing that's really different between them is the details of their affinity, mostly based, we think, on their interactions with phenylalanine 1759. There is a kind of block that is lipophilic. It is voltage independent, it's low affinity, and it has a similar value between a whole range of drugs that can exist in the channel in uncharged form. If you mutate any one or more amino acids, their affinity changes very little because they can make up the energy of interaction by just interacting with another amino acid or with the mutated amino acid side chains. Phenylalanine 1759 is the principal interaction that orchestrates high affinity voltage dependent action. It reduces voltage sensor block by greater than 100-fold, and for benzocaine by about 20-fold. It's important for me to emphasize to you right now that we don't think it's that the phenylalanine lidocaine energy of interaction is so great that it's this interaction and this interaction alone. And so I choose this word orchestrate very carefully to give you the idea that it's this phenylalanine interaction that sets up the position of lidocaine between domain 3 and domain 4 in such a way as to produce the voltage sensor interactions which are absolutely required for antiarrhythmic drug action. And use dependence, the thing that we need in order to get an antiarrhythmic drug, requires not only affecting the voltage sensors, but you have to do it with a time course that is very close to the time course of channel gating that exists in the physiological context, so that you can build up a block with pulses and be an effective antiarrhythmic drug. So let's see, how did I do? I did pretty well. And this is the group that was in the lab this summer. I would like to point out that we change on a regular basis. We work on a number of different projects, and Mike, who's the main person that I work on voltage-dependent sodium channels with, was not at the picnic where we went to because he was in Salt Lake City, and Harry was in North Carolina. But despite the fact that we're far apart, we continue to work together, and we hope to be able to bring this story to a final conclusion so that we understand the difference between each of the drugs that are clinically used and why it is they fail or succeed at being good therapeutic agents. Thank you very much.
Thank you, Dottie. Questions? Yeah, Stan.
Yeah, Dottie, I guess I haven't kept up with the sodium channel literature that closely, but I guess recently people have, who've looked at the issue of hypokalemic periodic paralysis, have wondered about the fact of whether, in fact, the voltage sensor itself actually goes through its own sort of pseudo-pore. And I'm wondering whether, since on the one hand we have to sort of, if we're looking at an agent binding within the pore, we have to assume some more diffuse effect to get actually at the gating particle. I'm wondering whether there may be any possibility that these drugs may actually be binding within this sort of pseudopore to give rise to the actual gating block.
So, well, there's like several levels of answers to that question. There are no data available to suggest that molecules of this size can fit into the little crevices that you're talking about. Although they're all canes, there are a wide variety of sizes and geometries of these various molecules. And so this gating pore that you're referring to is, first of all, not existent in wild-type channels. It occurs when usually a histidine, or often a histidine, is introduced that provides a pathway for protons and water. And in some cases you can make mutations that will permeate other small metal ions. There's no evidence that wild-type channels do this. There is in the cardiac sodium channel a little space that people think is behind the selectivity filter, whereby uncharged drugs or tertiary amines like lidocaine can escape. And there's some nice data, back in, I hate to say how long ago, a long time ago, we reported that in the cardiac channel you could block the cardiac channel with extracellular QX molecules. And then subsequent to that, Bill Catterall did some nice mutational data in which he found an amino acid that's different, that allows for access and egress to happen behind the selectivity pore, the pore helix. But those are, really tiny details compared to the rest of this. But you bring up the most interesting part of this whole thing, which is why on God's green earth can you bind something in the pore and affect the voltage sensors, which we know are way out there in the lipid bilayer. They're not anywhere close. And so clearly there is a link that occurs from through domain 5 in order to be able to produce this. And boy, we're really hoping NIH is going to let us play with that question for the foreseeable future, because we think there's lots of good evidence to help us understand where the energy of opening comes from and what the relationship of the position of the S6 helices has to do with the S4. But clearly they're related because the channel opens.
Okay, Dolly.
Yes.
I think I kind of lost track a little bit through the data, but what kept coming to my mind, and it relates to exactly what you just said, the use-dependent block is really an open channel block.
Yes.
So it's an enhancement of the block in the open state.
Mm-hmm.
So
the open inactivated state.
Okay, but
because we also have a paper in the literature, because if you disable the inactivation lid, lidocaine affinity goes way down. Okay. But if if you hold the voltage sensors up, we published this last year too.
Okay.
If you hold the voltage sensors up and you have an intact lid, you can achieve high affinity block
Okay.
at a
Okay.
negative potential.
Yeah, that fits exactly, I think. So, if it's, so, it could be that when it's in the open, it gets into the open state, the open inactivated state, whatever you choose it to be, and the affinity is higher, and that locks the voltage sensors
Yes,
in the up
exactly.
state.
It
And it
does.
doesn't require any physical interaction.
Absolutely.
To the
In
voltage
fact,
sensor. It's
that's
just-
what
and
it
that's
does
what's
is
seen
it
in K-channel.
it stabilizes the channel in the voltage sensor up state.
Yeah.
Of course, domain 4 is never really voltage sensor up, but it stabilizes domain 4 and wherever the hell
Yeah.
it is. And it stabilizes domain 3 up.
So, from your, I never saw the selectivity filter, so I'm assuming this is down near where the bundle crossing should be.
No, actually, phenylalanine, the selectivity filter, you know, it's about like that.
It's
It's
in,
underneath the selectivity filter, and in fact, well, the paper that I didn't show you any of those data, but if you put a positive charge here, you affect single channel conductance because of electrostatic interactions.
so it's high at the cavity ion site or into the
The
barium site?
tyrosine is at the bundle crossing. The second amino acid, the tyrosine.
So it goes right through the inner cavity?
It goes
and
right through in the open and activated
So this
state.
issue about where voltage dependence could arise without, so there is one additional thing. You said voltage dependence could arise because the blocker moves through the field.
Right, yeah.
Or the blocker affects parts of the protein that move through the field,
Right,
which is what you come down to.
right.
But it can also be, and this is what we lean towards maybe in some K channels with, for instance, polyamine block,
Uh-huh.
and many other blockers, of course, that they are affecting K-ion movement through the filter. So
Right.
that even though the blocker doesn't have to go into the filter and doesn't have to enter deeply into the field, if it obligatorily moves permeant ions in front of it, that's charge movement. So I'm wondering how much
Oh, we have
charge
no
movement could come
evidence
from
for
that.
that.
No, but, well, what about, okay, the evidence would come from transpermeant ion effects on the blocker potency.
Right.
And maybe transpermeant ion effects on the open channel block.
Yeah,
When the
and
positive charge
there, as far as I'm aware, we've tried, we did a bunch of experiments with these mutants of 1759K, and, you know, direction of which you go and which ions you use, and then it doesn't change. It's, in fact, for a long time, it really bothered me.
So this
But
make,
now
yeah, I mean, it
that-
makes me wonder why in K channels you have such strong transpermeant ion block,
Because in
transpermeant
K
ion effects
channels,
on inner cavity blockers
right,
like TA.
because in K channels, you have a very different kind of selectivity filter. At least we believe you have a very different. In K channels, you have this, you know, this, you really have a, you know, 5, 7, 4, we could argue how many angstroms.
Single channel filing, single
Right,
line
and
filing.
you really have this, you know, sort of problem. In sodium channels and calcium channels, probably, we don't have any structures, so this is, you know, but it really looks like, you know, there's
Yeah.
not this.
Do you not get much from the?
You really, it's like this pore kind of played out, and now you've got a side chain making the selectivity filter instead of carbonates. Kind of like a little bit like the KNA channel.
Yeah, that's right, so the
Right,
NAK
it has
channel
this
should go.
problem, it's on its way, because what it does is the top ones are turned out so that the side chains
Okay.
face, and probably sodium channels.
So then where's the field in that channel? You know, it's
God
easy to
knows.
argue that in the K channel, oh, it's
Right.
going down that chain of
Right.
K ions, but either it's on one ion here, which could mean all or nothing, and you could lose the trans ion effect, but you could still just pop that out and.
Well, except that you can measure voltage-dependent blockers in the channel. I mean, that's been done for, you know, a long time. So you can use cadmium block or, you know, QX block, 75% from the inside, 20, you know, from the inside of the.
And this is putting an amine right into that one spot, is it, in that case?
It's about 10 angstroms away, but it affects single channel conductance.
So the phenylalanine mutation, it's very interesting that you get rid of the use-dependent block, and also you get rid of the body sensor effect. So it looks like that that side is very important for the body sensor block movement. Is it possible that you actually have a gate kind of a gate and a charge coupling over there, so you kind of immobilize the charge movement when you have that phenylalanine and lidocaine in there.
Yeah, but you know, a negative charge would have done the same thing. We can put a negative charge at phenylalanine too, and it really does the same thing.
Yeah.
I mean, so
that means that does that mean that a lidocaine is not interacting with that site?
An amine is a sort of interesting chemical entity. It isn't like a frank positive charge. It's very protected by the hydrogens that are around it. And so I must admit, I'm not much of a chemist. And I predicted, wow, we're going to put a negative charge there and we're going to make it better, And then it wasn't better, and I had to go sit down, and Gregory had to give me a little, chemistry 101 lesson about the fact that it really, lidocaine is, there's something very special about the pi electron cloud that comes from the ring, and Dick Horn's data shows that very well as well.
Is it possible actually you have a lidocaine that is actually a large molecule, and when you get rid of this pi interaction, you are actually the two ends of lidocaine interaction are coupled, so that you need those two ends to talk to each other to get this charge immobilization.
Yeah.
And once you don't
I don't
have...
think it's the two ends. I think it's
so
the
it's
fact
the
that it has to wedge in that little space between domain 3 and domain 4. It turns out that the tyrosine 1766 has killed off several graduate students and postdocs in a number of laboratories, because you would really think that that's the really important guy, but that tyrosine is very, very hard. It's very hard to show much of. There's some very odd things that happen down there, and it's on the other hand, since we are going to have to affect, I appeal to simple logic. Since we're going to have to affect the voltage sensor movement in two domains, it's almost impossible to imagine that a single site interaction with one contact place is going to be sufficient to produce such a massive gating change. And someplace we have to get some cooperativity going, or we're never going to be anywhere. And we think that it's that lidocaine shelf that just sits in just the right place and is not as optimal for benzocaine, but can be very optimal.
You mean the leucine shelf?
The leucine in domain 3.
The leucine shelf.
Oh, the leucine shelf is very important for holding the amine, in that little crevice that now makes domain 3 and domain 4 marry together where they aren't normally. It provides some energy of interaction. And we have some experiments we think that are going to help show that, but we haven't done them yet. So I can't tell you about the way they turned out.
Now, I don't know if it's good or bad that you have 4 non-identical domains, because in K channels where you've got 4 identical ones, we hear these stories from Mike Sanguinetti about how these non-symmetric drugs sit in kind of awkward, non-central positions and can go around each
Right,
one.
and they
In
can
your
sit
case, you
in
can
the-
argue that it's lodged between 1 and 2 with an arm in 3 and a foot in 4. But
That's right. So on some days of the week, that's an advantage, and on other days of the week, it's a disadvantage. But clearly, from the point of view of how these drugs work, it's critical that they interact with domain 3 and domain 4, all of the channels, and I would say, all the anticonvulsants that interact with sodium channels also interact with these same amino acids in domain 3 and domain 4. So there's lots of drugs for which this is a sweet spot of drug channel interaction.
Are there any human mutations that are known that affect
Lidocaine sensitivity?
Yeah, and
Yeah.
causes people to die from cocaine?
So they're from cocaine. I don't know about the cocaine part.
Athletes
I'm the
that drop
research
dead
guy.
from a big snort or something.
But Robert Dumaine and Charlie Anselovich have a paper in Circ Research that came out earlier this year, in which they identified a patient patients that had mutations in voltage sensors, and in one case it's a voltage sensor, in the other case it's another related kind of part of the channel that produced very, very high affinity for lidocaine, and where the
Apparent
patient
affinity for lidocaine.
where the patient practically died when was given antiarrhythmic drugs, and because of the high sensitivity to them.
How does it fit with the model?
Well, we have a whole theory. I don't have time. I'll tell you over dinner if you want. We have a whole idea about how it makes sense. And
I was looking for a human mutation that
No.
suddenly appeared at the lidocaine binding site
yeah,
in
no,
S6.
no,
you haven't
these
done
are
that yet.
these are not. These are gating mutations that have indirect effects on the affinity.
So you can open a different order, because there's LGT3, and then there is persistent component of the sodium current.
Yeah. And then there's another renewal, and then I'll be explaining to students why 50 years after
So
the
why is
discovery of the
this,
drugs we can't.
So what's the difference in the structure of mexilidine compared to lidocaine, that one of them preferentially blocks the late component and another one the early component? Have you
Well,
thought about it?
I haven't looked at.
'Cause it's very
It's
interesting.
true that mexilidine does tend to block, but
The late
probably because of the match of time constants that
It's probably the
cause
first,
it to just block more slowly, and then it gets there finally.
So it's the timing during the gating.
It's all the timing. It's
It's
all
a good
the
assumption.
drug going on and the drug coming off, and it's. I think they all have the same, or charged drugs have very low affinity
It
for the
depends
for the.
when it happens, and when it happens,
when
the
it
channel will be in a
happens.
different state.
Right. Yeah. And how fast the channel's gating.
So it's possible you have a whole slew of drugs that have similar affinities, is what you're saying, I think, but their accessibility is altered,
there's
so they're slower on and off.
How
accessibility.
fast because then they basically operate on a different channel, right? Because now the occupancy of different channel states will be very different.
Yeah.
With
I think what this
But the
tells you
steady
is
state
that you
occupancy
have
could
to
be the same.
separate in your mind now. You can't get taken, too much by the specific experiment. You've got to look at the experimental protocol and ask yourself, is that ED50 they're reporting susceptible to voltage-dependent block, or is it less susceptible? And then can I understand how the drug is actually
And also,
working?
if you think about the binding in terms of time progression, that is on the order of the time of gating. or
Right. Mhm.
Then you can't think about the channel as a permanent structure. It's
No.
not a constant.
In fact, it's not a permanent structure. That's the
So
whole
maybe
reason.
that's a lot of the key of
Antiarrhythmic drugs.
why different drugs do different things.
Right.
at certain times.
Absolutely. But if they only have lipophilic block, then they're never
Then it
going to.
doesn't matter.
If they never interact with the voltage sensors, they're never going to be antiarrhythmic.
Being is the process of becoming, and getting there is half the battle.
Yes. Please put a good word in for us. Thank you very much.
All right, thanks.
You too. Thanks.